So dear students, today we are going to discuss about lac operon in details. First of all, it is a type of negative inducible operon. So the regulator will produce a repressor and usually the system will be in an off condition and must be turned on. And later we will discuss about the metabolism of lactose. Then we will discuss about the enzymes involved in lac operon. Then an inducer is also present, which is known as low lactose. And later, at the end of this session, we will discuss about mutations in lac repressor region, promoter and operator regions. Before we start, we have to introduce lac operon. It is a system which consists of many genes that includes promoter, operator and structural genes. And in addition to that, one more gene is there which regulate the function of this whole structure, that is the regulator gene. Structural genes codes for the main enzymes which are required for lactose metabolism. First one is LAC-Z, which encodes beta galactosidase, which breaks lactose into their monomeric units. Then LAC-Y, code for permease, a transport protein which allows or permits the passage of lactose from extracellular medium to the cytoplasm or to the inner side of the cell. And LAC-A is trans-acetylase. Then the repression of lac operon never completely shut down the transcription. This is an important point to note. This point I will explain it later. So how lactose metabolism inside E. coli cell, inside E. coli takes place? Okay, first of all, permease, a transport protein, allows the entry of lactose into the cell. So this activity is affected by LAC-Y gene. So extracellular lactose will be transported into the interior of the cell, where the enzyme beta galactosidase breaks lactose into galactose and glucose. In addition to that, beta galactosidase acts on lactose and converts them to a similar compound which is known as allolactose. So the function of beta galactosidase is to split lactose into galactose and glucose or to allolactose. Not or to allolactose, but it also produce allolactose. So this is how lactose metabolism takes place inside the cell. So here you can see how lac operon works. In the first case, lactose is absent. So there is no need of switching on structural genes for the metabolism of lactose. So you can expect a turned off condition of structural genes. So on the left side you can see regulator gene which are not a part of this operon but can regulate the function of structural genes by producing active regulator protein. Here it is a repressor, as lactose lac operon is a negative repressible operon. Negative means the regulator gene will produce a repressor. Inducible means it is usually in an off condition and must be turned on. So in the absence of lactose, active regulator protein, that is the active repressor, will be produced and it will binds to the operator region and blocks RNA polymerase. So there is no transcription. So in the presence of lactose, what will happen? In the presence of lactose, when lactose is present, then transcription and translation of regulator protein on the left side will produce an active repressor, but this active repressor binds with allolactose and this binding inactivates their activity, so it cannot bind to the operator region. So RNA polymerase can transcribe structural genes and functional products will be produced. So the functional products are beta-galactosidase, permease and transacetylase. So the question is how lactose will be transported into the cell. Even before the entry of lactose, a small amount of transcription will occur that will activate permease. So this allows the passage of lactose into the cell. When lactose is present, then enhanced activity of structural genes will follow. So this is the term which I want to explain, that the system is not fully shut down. പറയുന്ന പോയിന്റ് എന്താണെന്ന് വെച്ച് കഴിഞ്ഞാൽ, എങ്ങനെയാണ് പെർമിയേസ് ലാക്ടോസ് ഉണ്ടാകുമ്പോഴേ പെർമിയേസ് ഉണ്ടാവുള്ളൂ എന്ന് പറഞ്ഞു. അങ്ങനെയാണെങ്കിൽ ആദ്യത്തെ ലാക്ടോസിനെ അകത്തേക്ക് കടത്താൻ വേണ്ടിയിട്ട് ആര് സഹായിക്കുന്നു? കാരണം ലാക്ടോസ് വരുമ്പോഴേ പെർമിയേസ് ആക്ടിവേറ്റ് ആവുകയുള്ളൂ. അപ്പോൾ എങ്ങനെ ഫസ്റ്റ് ലാക്ടോസിനെ അകത്തേക്ക് കടത്തും? അതിനുള്ള ഉത്തരമാണ് ദിസ് operon system is not fully shut down. There is a slight transcription of structural genes that will allow the activity of permease and can allow their transport across the membrane. So lactose, in the presence of lactose, this is how lac operon works. This is how the organization of promoter, operator and structural genes in a lac operon. So you can see the site which are represented in yellow is lac promoter region, where you can see -10 region, -35 region. So minus sign indicates from transcription starting site the bases can be numbered -1, -2 onto the left. So this region is -10 region is which consists of bases 10 away from the transcription site on left. And you can see the lac repressor, the product of regulator gene, will binds to the promoter region and overlaps the some of the structural genes too. So this is how repressor blocks RNA polymerase. It binds to the operator region, but actually it binds to some part of promoter as well as structural genes too. So this is how it works. Now we are going to discuss about lac mutations. Before that, we have to introduce one term, partial diploid. Throughout this lecture, we will use the term partial diploid. That's why I'm going to explain this. Partial diploid is a full bacterial chromosome plus an extra piece of DNA on F plasmid. So this is a partial diploid. So now we are going to explain what happens if structural genes gets mutated. So first of all, structural gene mutation will affect the structure of enzyme but not their regulation of synthesis. So now we are going to represent a partial diploid. Here the plus sign indicates there is no mutation and minus sign indicates mutation. So in this partial diploid you can see that lac Z is plus, lac Y is minus, lac Z minus, and the counterpart of that particular partial diploid, lac Z is mutated, lac Y plus. Here at least one copy of DNA fragment is enough to produce a functional beta galactosidase and permease. Because you can see that on the left side, lac Z plus is positive. That means that is enough to produce beta galactosidase. Even though on the right side you can see it's the partial diploid part of this particular gene is mutated. Even though it got mutated, there is no such problem in producing beta galactosidase. So one copy is enough to produce a functional gene. Same is the case with permease 2. So on the left side you can see lac Y minus. That means that gene code for permease is mutated. So on the other half, lac Y is positive. That means that is enough to produce permease. Now we are going to explain what will happen when regulator gene get mutated. So here the point to note is lac I minus means lac mutation leads always constitute transcription of structural genes. So if regulator gene get mutated, then structural genes will be transcribed constitutively. And one more thing is there, lac I plus, that means a normal copy of regulator gene is dominant over lac I minus and is trans-acting. A single copy of lac I plus brings about normal regulation of lac operon. So if you represent lac I plus bar lac I minus, here also you can expect a fully functional regulator gene product, because lac I plus normal copy is dominant over mutated one. So and is transacting, that means it can act not on this part but can act as like a transacting activity is there. So in the partial diploid, lac I plus, lac Z minus, lac I minus, lac Z plus produce fully functional beta galactosidase, because it's lac I plus is dominant over lac I minus. One copy of lac Z is mutated, but the other copy is fully functional. So this will be the final product. Functional beta galactosidase will be produced. Now let us look at what happens if lactose is present and what are the problems associated with mutations at regulator, promoter and operator regions. First of all, the in the absence of lactose, you can see the representation of partial diploid on the left side. P1 lac I plus and lac I minus. Here you can expect an active repressor from lac I plus and a mutant repressor from lac I minus. As I told you earlier that lac I plus is trans-acting, so it can act on both partial diploids on the left side, on the right side I think. So here on the right side you can see lac O promoter region is okay, it's not got mutated, and lac O plus, that means operator region is also fine, and lac Z minus on the upper side, and at the bottom of this you can see lac Z plus. On the upper side it's lac Z is mutated, but on the lower side lac Z is fully functional. So what will be the product? So here from regulator, active repressor will be produced. It can act on both operator regions and can prevent transcription. So there is no gene product will be produced from the from this case. So lac I plus gene is transdominant. The repressor produced by lac I plus can bind to both operator and repress transcription in the absence of lactose. So what will happen when lactose is present? Then okay, then the same is the condition on the left side. Lac I, one copy of lac regulator gene is active, the other copy is mutated. So when lactose is present, allolactose will be present. It inactivates one repressor, so repressor cannot bind to the operator region. So on the upper side you can expect no transcription and translation and non-functional beta galactosidase because lac Z here is mutated. So on the lower side you can see that lac O P is okay, lac O lac P is okay, lac O is also okay. Then comes the structural gene lac Z. It is also in a functional condition, it is not mutated, so you can expect transcription and translation. Why? Because here in the case of in the presence of lactose, active repressor got inactivated. So this is how lac operon works in the presence and absence of lactose when some of the regions of gene got mutated. Now let us look at another mutation that is known as super repressor formation. So sometimes lac I will undergo a super mutation that will produce a super repressor. The problem with this super repressor is that it does not bind with lactose. So even if lactose is present or not, it will not bind with lactose. So this representation, this partial diploid representation, you can see that lac I super mutation caused super repressor formation. On the bottom you can see lac I plus, which also produce active repressor. So this actually binds with lactose, but super repressor will not be affected by lactose. So lac P, lac O plus, lac Z plus on the upper side will be affected by super repressor. So it binds to the operator region and inhibit RNA polymerase. On the lower side also you can see that super repressor binds to the operator region and also blocks RNA polymerase. So in both cases lack Z plus genes, that means beta galactosidase will not be formed. So this is how super repressor works. So the next is lack operator mutation. Lack operator mutation can be represented as lack OC, which C represents constitutive, always expressing, and lack O mutation is dominant over lack O plus, which is cis acting. So in this case, lac I plus, lac O plus, lac Z minus, lac I plus, lac O constitutive, lac Z plus produce fully functional beta galactosidase constitutively. So now we are going to discuss about what will happen to the partial diploid lac I plus, O plus, Z minus, I plus, O constitutive, lac Z plus. In the absence of lactose, lac I plus will produce active repressor. It will try to binds to operator region. One region is okay, that is lac O plus. So there we can expect active repressor binding. So at that region no functional gene will be produced. No functional gene will be produced because lac O plus is already occupied by the active repressor, so it will block RNA polymerase. And one more thing is there here, lac Z minus is also mutated, so non-functional beta galactosidase will be produced only when lac O plus is constitutive. So in the first case you cannot expect a functional or the synthesis of beta galactosidase. On the second region, what will happen is that active repressor will try to bind to the operator region, but the operator region here is mutated, so it cannot bind to the operator region. Here RNA polymerase cannot be blocked, so it will transcribe. Lac Z is also positive, so it will produce a functional beta galactosidase. In the second stage in the first stage itself, when lactose is present, then active repressor will be produced. This active repressor binds with lactose to form an inactive repressor. So inactive repressor will not bind to the operator region. So at the first site, lac O is positive, lac Z is minus. That means RNA polymerase will transcribe and produce a non-functional beta galactosidase because lac Z here is mutated. On the next side, on the other hand, inactive repressor will not bind to the lac OC. It is already mutated, but inactive repressor is also present, but it cannot bind to the operator region, so transcription occurs. Here lac Z is plus, that means transcription and translation will produce functional beta galactosidase. The next example is partial diploid lac I plus O plus Z plus I plus OC lac Z minus. In the absence of lactose, active repressor will be produced by the regulator gene. Here lac O is plus, lac O on the other hand it is constitutive, it is mutated. So what will happen is that in the first stage lac active repressor binds to the operator region and blocks RNA polymerase, so no beta galactosidase will be produced. On the other hand, here lac I plus is transacting, so it will try to bind to the operator region here, but the operator region is mutated, so it cannot bind to the operator region. And but the problem is lac Z is a mutated version, so it cannot produce a fully functional beta galactosidase. So transcription and translation will always produce a non-functional beta galactosidase. So in the presence of lactose, what will happen? Here lac I plus always produce an active repressor. It binds with the lactose and inactivate the repressor, so repressor cannot bind to the operator region on both cases. So on the upper side, lac P, lac O plus lac Z plus will always produce a functional beta galactosidase, but on the lower side the problem is lac Z gene is mutated, so transcription and translation will produce only non-functional beta galactosidase. So now to the last section of mutation in lac operon, promoter mutations. Lac represented as lac P minus, it's cis-acting. Lac I plus, P minus, Z plus, I plus, P plus, Z minus fails to produce a functional beta galactosidase. Because what will be the problem? Lac P in one case, in one fragment of partial diploid is mutated, so it cannot produce, as it is a cis-acting one, it cannot produce a functional beta galactosidase. Now to the conclusion of mutations. First of all, structural gene mutation, location lac Z, Y, only affect lac Z or lac Y. The effect will be alter amino acid sequence of protein encoded by gene in which mutation occurs. The second one is regulator gene mutation, I, which is transacting, affect transcription of structural genes. Operator mutation, lac O, cis-acting, affect transcription of structural genes. Promoter mutation, lac P, cis-acting, affect transcription of structural genes. So when we conclude this section, you can see that any mutations, whether it is happening to regulator gene, operator, promoter, that will always affect transcription of structural genes. So this is about the conclusion of lac mutations.